Last week, I introduced my mini-series, Autism Genetics 101, by familiarizing you with the different types of genetic variations or mutations that are sometimes found in the human genome. If you haven't watched that video, I highly recommend starting there and then coming back to this one so you're more familiar with the terms I'm going to be using. Okay, so first, a little history lesson. Up until the 1960s, Freudian psychoanalysis still had a pretty firm stranglehold on professional perceptions of autism. In 1964, Bernard Rimland published his book Infantile Autism, which squarely placed autism's etiology into the realm of biology. So with this work, we thankfully left behind the era of the refrigerator mother, which was the idea that emotionally cold and unresponsive mothers were at fault for their child's autism or schizophrenia. By the late 1970s, that's when we see the study of autism genetics and heritability in its infancy. Susan Folstein and Michael Rutter published their work on autism concordance in identical and non-identical twins. Now, concordance simply means that each twin in a twin pair is diagnosed with autism. If one has autism and the other doesn't, then they're considered discordant. Anyways, in Folstein and Rutter's initial study, which was relatively small, they found that more than 1/3 of identical twins were concordant for autism. But by the time we get to larger twin studies done in the 1990s, we see that figure shoot up to 60 to 90%. The 60% refers to an exact diagnostic match between twins, so autistic disorder and autistic disorder, whereas the 90% includes twin pairs that maybe didn't match exactly diagnostically, like one had autism and the other had Asperger's, or maybe one had autism and the other had a broader autism phenotype. Meanwhile, these studies showed that non-identical twins have concordance rates similar to what we see in non-twin siblings, which usually runs about 12 to 20%. Now, during this early genetics research, scientists were initially expecting that autism might be a single gene condition, which made more sense when you consider the old 1 in 10,000 autism prevalence rates of the 1970s and the 80s, because usually the rarer a condition is, the simpler its root cause tends to be. However, what with the broader autism phenotype that scientists were seeing pop up in twin studies in the 1990s, it was becoming clear that autism was not as genetically simple as they'd once hoped. Also by the 1970s, reports were coming out linking high rates of autism in association with maternal infections like rubella. And then by the 1980s and 90s, other reports were linking autism with conditions like Fragile X syndrome and tuberous sclerosis. This growing evidence continued to lend weight to the idea that autism was genetically heterogeneous, meaning that it likely had many genetic causes. And since that time and the sequencing of the human genome in 2003, scientists have identified hundreds of genes with major or strong effects on autism susceptibility and perhaps thousands of genes with more minor effects. In short, the genetics of autism are complicated, and at least from that perspective, each person probably carries their own brand, so to speak. It reminds me of the phrase, just because you've met one person with autism, you've only met one person with autism. Well, the same kind of goes at the genetic level too. Now, what kind of genetic variants or mutations do we tend to see in autism? Well, for one, we know that about 5% of people on the autism spectrum have major chromosomal abnormalities, the type you can visibly see on a karyotype. People with these genetic variants are also probably going to have some kind of syndrome with other symptoms or medical conditions along with their autism, because many genes are likely to be affected. People with these conditions may also be more prone to having intellectual disability as well. Now, the next type of mutation that we see are copy number variants. These tend to be deletions or duplications of genetic material that usually range from about 1,000 base pairs up to 5 million nucleotides long. The larger they are, the more likely they're going to encapsulate multiple genes. So again, people with these types of genetic variants are liable to have other medical issues alongside their autism. And like the large chromosomal abnormalities, these folks are also more likely to have intellectual disability. Now, about 10% of people on the autism spectrum have new or otherwise known as de novo point mutations. These mutations tend to be relatively rare. And meanwhile, another 10% have other types of rare variants that we haven't already covered here. Now, finally, it's estimated that about 40 to 60% of autism susceptibility is controlled by common genetic variants. These are variants that happen in at least 1 to 5% of the population, if not more. This type of susceptibility is probably also polygenic or multi-gene in nature, meaning that a single common variant isn't likely to lead to autism all by itself, but when partnered with other factors, autism is more likely. As you can probably guess, genetic susceptibility factors for most people on the spectrum are probably going to fall into this last category. Unfortunately, because these genetic variants are common, it can be really hard to determine if one in particular is playing a measurable role in susceptibility, because statistically, it's hard to identify them without studying huge numbers of people. And by huge, I mean as much as possibly hundreds of thousands of people. So you can probably understand why we're really behind in identifying and understanding these common factors. While common variants are still sort of the next frontier in autism genetics research, what do we know about the rare ones? Are there any commonalities in terms of what these genes do in the cell? That's an excellent question, and in fact, there are. So when we study major effect genes, i.e. genes that when mutated are very likely to lead to autism, we can see that a large number of them are active inside the nucleus of the cell and help to orchestrate the expression of other genes. A lot of them are kind of like master regulators, affecting the expression of a lot of neural-related genes. Now, another major category of autism genes are those that function at the synapse, which is the point at which neurons meet and communicate with each other. Some of these genes produce proteins that help to stabilize the synapse, while others are involved in the synapse communication itself, like receptors for certain neurotransmitters. Now, if you've watched my earlier video on the Cambrian explosion of autism genes, you probably remember that a lot of these major effect autism genes evolved all around the same time in history during the Cambrian period in some of our earliest fish ancestors. This was also the point in evolutionary time when the first true three-part brains evolved. Before that, we had centralized nervous systems, but not what we would consider true brains. As such, a lot of autism genes that evolved during that time, thanks to a couple of rounds of whole genome duplication, they're probably really important in expanding the variety of different neuron types we see in vertebrates today, as well as expanding the complexity of the landscape of the brain itself. A lot of these epigenetic regulators in particular were probably pretty handy in that fascinating nervous system redesign. Now, all that being said, the prevailing research also suggests that environment can play roles in autism susceptibility. For instance, research has shown that prenatal environmental factors like infections, exposure to medications like anti-seizure meds, prenatal exposure to alcohol, as well as folate deficiency all share significant links with autism. So there's probably a lot of fascinating and complex gene/environmental interactions potentially going on there. And in fact, I think that's kind of cool because it suggests that these developmental networks have a certain amount of plasticity or ability to adapt in the face of environmental influences, which can be a good thing in evolutionary terms. After all, genetics is definitely the blueprint, but each gene can often be produced in a myriad of number of different forms, especially neural-related genes. So that's an exceptional amount of potential plasticity there. Anyways, thanks again for following me down this genetics rabbit hole. Please leave your comments and questions in the comment section, and I'll definitely get back with you. Thanks again, and until next week, ciao.